Thank you very much, Dr. Janaki, for your very valuable and impressive lecture on microbiome and also the techniques used to make genetic arrays of microbiome. So now the lecture is open to discussion. We welcome your queries, questions, problems in microbiome. I mean, to start with that, I mean, you said that the difference of microbiome in naturally born babies and cesared babies, and also the food we used to eat a few decades back and the food we eat now. How do you explain, I mean, it is not very fair to ask from a scientist about speculations, but your gut feeling, the changes we are taking place now, even here, that the rapid changes in food habits and also the other health behavior. What can you just tell about your gut feeling?
Because you use the word gut feeling, I guess it comes from the microbiome in the gut. We know that there is a connection between the gut and the brain as well. So there are lots of scientists studying that. I don't know the outcome yet, but even we are starting to do that. So there is C-section has saved babies. Everybody knows, right? But if there is a need to do that, you have to do that. But you have to keep in mind that the naturally born babies, when they pass through at this moment we believe that they are sterile. So babies are born sterile because in the mother's sac. And once they come through the birth canal, we believe that they get their first dose of microbes. And that stays for life. So that's very important. And like I mentioned, the sialylated oligosaccharide that is produced in the breast milk is only digested by Bifidobacterium longum infantis. So the question, a lot of people are working on that, and the question is, why would a mother produce something to feed the bacteria, not her own baby? And when you do a C-section, we all know that the babies get antibiotics at the beginning. So their microbiome has to be totally different. People are working on that, and people know that they have different communities, but I do not know at this point if they've been able to link the cesarean born babies and the naturally born babies who's prone to diseases and some. But I know there are some studies that shows these babies have more allergies and asthma and stuff like that. We don't work on that yet. We are doing a lot of basic microbiology. The food. So as we all can imagine, the bacteria are unable to digest or get anything out of processed food. Since they were not, they didn't evolve to do that. They can digest, synthesize vitamin K, synthesize short chain fatty acids from natural food. But of course, the world is changing. Maybe bacteria have changed too, but we don't know the answer yet.
So those days, scientists considered breast milk as the wholesome food.
Yes.
Not looking at the microbiome, they look at the nutritional components. But now this statement has more value.
Yes, certainly. Certainly. So they encourage, in the US, they encourage people to feed babies with breast milk as long as you can. And I don't know how familiar you are with the vaginal microbiota. So there are groups of mothers who are willing to eat the placenta of the mother after the baby is born because they think that is the most nutritious thing and that you should not throw it out. And that encourages good bacteria to colonize your baby. And then there are also the scientists, some of them I know, that they take the fecal material from the mother when the baby is born and they rub it on the baby so that the baby gets the good bacteria from the mother. Because we still don't know exactly how we get, but we know that we get all our bacteria from the environment, and the diet helps shift the communities. But we also carry the bacteria that were established in early life throughout your adult life. So there is a certain percentage that we carry throughout our lives, but then we get the other bacteria. And also those bacteria are prone to depending on the diet you eat, the Western diet. We do a lot of experiments with the Western diet and other different diets. But these are all done in mice. If you think about it, the human microbes are not their natural microbiota, but there's no other way to do these experiments to make.
Maybe you said that stress also play a role in that particular community that you said that
the gut feeling.
Okay, any questions?
You were talking about prebiotics. So how do we know whether we are promoting the development of the good bacteria, because we have so much of pathogenic bacteria as well? So what sort of food should we take in order to promote the good bacteria and not the others?
First of all, what I mentioned, didn't say in the talk, that when you compare the good bacteria and the bad bacteria or the pathogens, it's very few pathogens that cause disease. It's just like in the world, the good people versus bad people, it's very few. But the few very bad people or the very bad bacteria get the most attention, because we have to find ways to fight diseases. They appear from time to time during diseases time. So the word bacteria to the general population is all germs. But if you go by numbers, the number of good bacteria in our body is way more than the number of bad bacteria. So if you maintain your microbiome, you should not be able to, actually it's stopping pathogens from entering the body. The prebiotics, there are a lot of groups in the Western world, they study certain diets or certain ingredients just because they are interested in it, like broccoli, inulin, but they are not interested in our kind of food. So they don't do any research, but that does not mean that they are not good, that you should eat always kale or broccoli or everything that comes from the West. That doesn't mean that. What I think is that if you eat a good diet, a colorful diet, that should do. You don't have to eat all the processed food. I mean, I know there's no way you cannot. You should, you need it to eat it from time to time, but there are other ways to digest that food. But to promote health and to promote the microbiome, the good bacteria.
Okay, now they say eat the rainbow.
Yes, eat the rainbow. That's the way to go. Don't eat tablets.
Okay. Pass it.
A kind of patient actually who is really suffering with gastritis. So then I thought, like, now in Sri Lanka actually, when they diagnose gastritis, they usually go for like the endoscopy, things like that. So is that the same in those developed countries? Because as you said, the enhancement of good bacteria could cause really a better results than rather than medical drugs. They usually give us the various kinds of anti-proton pumps and omeprazoles, all kinds of things. But is there any other technique really to identify that whether you are really with good set of bacteria or that kind of diagnosis really?
First of all, I'm not a physician, so I can't really give you any.
Because all the time they go for medical drugs actually. So as you said, prebiotics.
Are there any physicians in the audience?
Yeah. Probiotics.
Master Helicobacter Pylori, I think you can.
Yeah, that is also actually tested in Sri Lanka. But as you said now, they consider it as a carcinogenic one. But that what you said was not really happening in Sri Lanka. It's to be considered it as a good bacteria.
So gastritis is a loose term. It's any inflammation. It's like the headache. Any inflammation, you don't know the cause, you just take Panadol. So just like that, you take the proton pump inhibitor because the acid. So what people, Helicobacter is one of the causes of gastritis. But like I said, we did some work with the microbiota, and we see that the mice do response by having there is an IgA response. So it's not like that, oh, these are good bacteria, I don't do anything. The host does something. But that's a very low level of an IgA response, big enough, but very low, to keep the bacteria, the good bacteria, in balance so that it doesn't kill them. So Helicobacter pylori is now it's a controversial topic because it's already in the Western, it's treated with antibiotics and it's removed. And like you said, yeah, if it gets worse, you do endoscopy. But when you just go to the doctor, if it is gastritis, they'll try different things, except maybe changing the diet and the lifestyle. So changing the diet and the lifestyle should be the first one. But there too, they try to remove the bacteria. So you can do a culture and see if you carry Helicobacter pylori. But it's not that every Helicobacter pylori is bad for you. And what they have seen is that in epidemiological studies, that removing Helicobacter, people who don't carry Helicobacter or removing Helicobacter right now, what's happening in the Western world, there's more upper gastric diseases. So that's the question we have now, whether it was a wise thing to call it a carcinogen and treat it with a triple regime of antibiotics, which is really bad for your good bacteria.
Any more ideas?
Isn't it a decoction now? You're
Get the
concentrating only
mic.
on bacteria.
Mm-hmm.
What about the other organisms?
There are a lot of other groups that are working on them. So phages and fungi, and they think that's the microbiome. We think the bacteria is the microbiome.
Combination of all which is
Yes. So there are a lot of collaborations going on even in our lab right now to see if bacteriophage does something. But one of the, we have to develop techniques to study all that. So right now we are only working with bacteria because that we isolate them and we can do all kinds of different in vitro assays with them and understand them. But there are a lot of other labs that do work with fungi and phage. And I only talked about the microbiome in the GI tract, but there are science groups studying the skin microbiota, and they have shown that your nose carries a microbiota. There's a microbiota on your right hand cheek and the left cheek, and so it's all about the entire body, which we have failed to understand before because we thought, okay, we are all humans and we carry humans and we take antibiotics and kill our bacteria, but the question is, are we microbial or are we human?
Yes, pass the microphone.
I am interested in a different way, but I am not going to ask the protocol or whatever it is. How that 16S rRNA that you have studied the whole microbiome, no, that in the system, and then first you screen the whole microbes which is available in the sample.
Yeah.
So that you basically the 16S RNA, the whole genome that you have done, what's a NGS, so what sort of thing that where you didn't get the bacterial isolate, no, the whole microbiome, the DNA that you have gone.
Yeah.
That's that based on the NGS. So what type of this thing that how do you analyze the 16S rRNA?
So as we all know that I'm sure you are familiar with the 16S ribosomal RNA gene that we carry
that is,
through
if
evolution.
you are going for the bacterial isolate, definitely the 16S rRNA that you have focused on the particular bacteria.
Yes.
So initially when you screen it, the whole microbiomes, that how did you go for the 16S RNA? That because it will be like all the specific region, the variable region is common to the most of the bacteria. Then it won't. It's difficult to you to sort it out the microbial community. So I just wondering that's the NGS technique that you all have gone, or just
We have developed our own technique in the lab, which we call it the CopperSeq. It
Okay.
stands for, I forget exactly now, but what we do is we extract the DNA.
yes.
It's a mixture of all bacteria,
Yeah.
fungi, everything that's in the
Yeah,
sample.
Yeah.
But then we have the universal primer specific for bacteria, right?
Yeah,
So we amplify the bacteria DNA only.
Yeah.
And then we have a program, we have programs
That's
people use
you
all
maybe
the...
a different technique to
Yeah.
sort it out, because
Yeah.
when you go for the 16S rRNA, that's for the specific, even the universal primer. So all the bacteria is mostly more or less little bit different with the variable region.
Yeah.
So that all will get amplified, and
Yes.
to separate that
Yeah.
it's a difficult. So nowadays that NGS and all is going
Yeah,
on. So
we-
that's
yeah,
what
we-
I'm
we-
interested
yeah,
to
we-
know that. So once it come that you are going for the specific media, and then you are amplifying the bacterial isolate, no? The
Yes,
specific
yeah,
bacterial
yeah.
isolate.
Yes.
Then with the from the media only you are separating, no? That,
Yes,
and you are going for PCR, that specific primers as well?
No, we just do the PCR
Okay.
and then it's like getting an overview of what's there.
Okay.
We may miss some things because we don't know how deep we have to go to get
Okay.
everything that's in the sample, because some of the abundant DNA
Yeah.
would actually get amplified more than the
Yeah.
less abundant
Yes.
ones. So once we get that, we have an idea of what's most abundant and what's less abundant. Sometimes we don't even use that sample. Just looking at it, we see that it's not worth going after it. But most of the time we do that to compare between lean and obese people. So we have a twin registry that we get samples from twins born and separated from, as adults, they live their own lives, right? So, but they still come back and give us samples over time. So you can't just look at one sample. You have to look at several samples over time. So we have lots of samples, and we have freezers and freezers full of all these samples. And then you can compare between these obese twins and lean twins. And the other beautiful thing about with these red libraries is, say you want to study a certain group of bacteria only, that's about, someday you realize, okay, this is, you suspect that this organism is the cause of colon cancer or IBD or something. You can go back to your red library, you can pick that well, and you can pick that species from all different donors that we have already made libraries. Samples collected from different geographical regions, like from Asia, from Europe, from US, and from different parts of US, eating different diets, and then compare between the obese and see what the bacteria are doing. Because now we have realized that even within a species, different strains are different.
Yeah.
So there are lots of strain differences, and so even if we see the whole plate is just full of, for example, E. coli, it's not the same E. coli in A1 versus 12H. So we try not to throw them, but we end up getting lots and lots of these kind of... So the other thing we do, which I didn't mention in my talk, is that we do whole genome sequencing of each organism that we have purified in the end.
Yeah, that's correct.
Yeah, and then we can compare, and then we can understand. But before we do that, we have to make our media to get them out, to culture them. At that point, we don't have that much information about the bacteria that are in there. Some of the bacteria species that we get, nobody has even published anything, so we just have to...
How many species
Okay, thank you.
are there altogether in the range?
A good question, I don't know.
Millions, is it? How do you keep track of all those? Because it must be a huge, the number of strains, the variation.
So from a single fecal sample, which, like I mentioned, it's about 0.5 grams. As you know, the entire fecal sample is not the same, right? It depends. So we can't go back to a second fecal sample because it's not reproducible. And so when we do that clarification step, we make enough stocks to last several years. And then we can go back to the second tube and get the same things out. But sometimes they even die between freeze thawing. And then we try to, we decide how many plates we are going to do. So right now, I believe there are like maybe less than, I should give you a number, 100, 200 different species so far. But each species has different, we get different strains because it's like a snapshot at that moment what you get, right? And there are a lot of genes being horizontal gene transfer, genes being transferred. So it's...
Antigenic variation.
Yes. But for each donor, we keep the plates. So we usually do like 5-10 plates so that we have 500 to 1,000 strains. And then try to build our communities and take what we want and use them for different in vitro assays, like collect all the same species or make communities and things like that.
Yes, Professor.
Yes, Janaki, I realized that one of the sources of prebiota is from the plants. Am I correct?
Yes.
Plant leaves like things. So are there any studies done to look at the direct relationship between the availability of some of these bacterial species on leaf and passing them on to the human beings? And at the same time, suppose if there's such a relationship, we know that many a time there are a lot of agrochemicals are being used on the plants. Suppose if they are being deprived, I mean if we eliminate such microbiome from the plants, I think that deprive the human being getting them in. So what do you think about this?
Yes, but so far the experiments we do are with germ-free animals, so we know exactly what goes into the animals, and so we give them sterile food. So in our experiments, they don't get into the system. But you're right, the food itself carries a lot of bacterial DNA. That's why I said, just looking at the 16S DNA sequences in the initial sample, you see bacteria, dead bacteria, live bacteria, and bacteria from the food itself. Sometimes these bacteria get into the mice and stay in the mice or in our assays, and at some point or at later steps also we can identify them. So they get there. But by cooking and all these methods, we kill most of the bacteria that are already on the plants. I think there are a lot of groups studying that, but we don't do that. We use high techniques.
Any more questions? It's a long, continuing discussion.
Can I ask something, if that's okay?
Yeah, why not?
A little bit, not a question actually, some advice for the young staff that we have here. Now you have gone a long way. I mean, you're working with the leading scientists working on the human microbiome. What advice would you have for the young staff here who are aspiring to be somewhere you are? I'm sure it's a little bit more than luck. So what advice what qualities, capabilities should they develop?
Like I said, I started on my master's with Janaki, yes, so maybe that's the way to go. We have to actually do a lot of basic. So we've got all high techniques in the lab. So even the students that come into the lab, they right away use the robots, right away extract DNA with using robots, and most students don't even know how to do phenol chloroform extractions and PCR setting up. They're all done with. I think we have to do a lot of basic research to be able to get there, because for one thing, when you have the bacteria on a plate and when we do growth curves, you see that they don't all grow at the same rate at the same time. So those are all basic things. So maybe sometimes they produce certain things in the log phase that they don't produce in the stationary phase, but there is no way we can wait for this to come up and this other bacteria. So I think it's good to start with a lot of basic questions and learn all the basic things so that we can. One of the important questions is, how do we build a community? So we know that we get the bacteria in early life, but after that it's the host who decides what we can take. So those things are very important, how you build a community and how, so a lot of basic bacteriology.
So it's a long discussion, I suppose. I mean, in the end, I mean, what we see is, we thought that after sequencing human genome, we achieved. But now I think the more complicated human microbiome, and also, as you said, that not only the species diversity, the strains, even we know that in malaria, maybe the species is same, but the virulence mostly depends on the strain. So I think that bacteria is concerned, it's much more worse. So it's huge area to study. And finally, to wind up the session, that I don't know, it is fair enough to ask this question, but nowadays there's a trend that people practice 8-hour feeding and 16-hour starving. They say that it is very good for your diabetics and all control that enhance immunity and so many things. You fed your sterile mice that you must have used different feeding patterns. Do you have any idea about the microbiome inside with the feeding pattern or feeding times and all?
We studied a group of humans who actually measure what they eat and do all kinds of this kind of stuff. I don't think we saw anything that is like, oh wow, you have to starve to be able to. I don't think so. And I don't know if starving is is the way to go. I'm not sure. No.
So anyway,
But when we do mouse colonization experiments, it's very difficult to colonize them several times. So they do. I think the students only do, because it's a lot of work. They do only once and see what we get. So we don't really starve them, and then because the animal rights, they have lots of rules, and we are not allowed to do that.
so anyway, till 1960s, mid-60s, we developed a lot of things to kill germ.
Yes.
Kill germ and microbes and, stronger and strong things. But now we should believe coexistence.
Yes. And treat them nicely. Kindly.
I'm here to do the formal vote of thanks on behalf of the Director, the International Relations Unit, Professor Rajendra, and the staff, and Director and the staff of IT. So this distinguished lecture series we organize in collaboration with IT and International Relations Unit and the SETMI. So the first lecture of the fourth series we concluded just before. Very impressive, interesting topic, and also a very fruitful, lengthy discussion. We really thank, on behalf of the Open University of Sri Lanka, to Dr. Janaki Lelwala Guruge and her beloved husband, our friend Bandil Guruge. So came all the way from Washington to Sri Lanka, and we got the chance to have her here to share her long years of research experience and the huge knowledge base. Bit of that knowledge base she shared with us. We on behalf of the Open University at large, we all thank you, Janaki, for your spending your valuable time midst of all your fun activities during last couple of weeks, that in fact you had to make your slides here and there when you're going here and there, Yala, and so you had to prepare. You forgot to bring your pen. So we all thank for your very valuable exercise and your very valuable talk. We'll give a round of applause to appreciate her work. We wish you all the best. Thank you.